Today we're going to talk about non-Mendelian inheritance. So far to this unit, we've talked about types of inheritance that are pretty predictable and ones that follow the law of dominance. So some laws, or excuse me, some traits are not going to follow that pattern. And the ones we're going to look at today are two examples of those. So for example, when we look at these flowers over here, the big thing to notice is even though they do follow a different type of inheritance, the law of segregation still applies, meaning that each parent can only give one allele of the two that it has. So Punnett squares are still going to work real nice for this. So if we look at this here, we have um, incomplete dominance as the first one. Incomplete dominance is going to be an example where neither allele is going to be dominant over the other. That's why we say incomplete. Instead, it's going to be a blend. And it's kind of hard to see here because I think this one looks kind of pink anyway. But this flower is red and this flower is white. So what that means is the heterozygous or the intermediate phenotype is going to be a blend of the two, a little bit of pink and red. It's like if you put paint together. So if you have the heterozygous individual here, you're going to see that this one is going to be a pink, a blend. So go ahead and look down here. I want to highlight some things here. If you look at here, you'll see that in order to be red, this one right here has to have two big R's. So two big R's are going to give you that red allele, or excuse me, phenotype. But if you have a big R and a little r, that's going to make you pink. I'll just put a P there. And then if you are little, that's going to make you a white flower. Okay. So kind of a neat twist on what we've seen in the past where there's really no dominance. It's going to be something that's going to do something completely different. So because of that, we're going to use different letters to represent the alleles. So for example, if we look back on this here, incomplete dominance, if we have this type here in the past, we've always seen this 1, 2, 1, but where in the other ones would be 3 to 1 as far as the phenotype goes. But because it's a blend, it's going to be a 1, 2, 1. So all that to say, the genotypic ratio and the phenotypic ratio is going to be exactly the same. Another example would be codominance. And codominance, that's where instead of neither dominates the other, this one is similar to that, but both get expressed. So instead of it being pink, it's going to be white and red. So when we look at this here, we can see that the flower is red sometimes on the petals and white sometimes on the petal. It's not a blend. So the heterozygous individual is going to be both are going to be shown completely. We're going to look at some examples of these in a second. The last one that we talked about is the variations of a variation on Mendel's laws. Has to do with how the environment is going to influence. So for example, these are identical twins right here. So you would think that everything is identical in their DNA and their phenotypes too. But that's not always true because they can have the environment change them. We don't talk about this one as much, but if somebody was exposed to more sunlight, they would be, of course, have darker melanin, but it could increase their chances of diseases too. This one's hard to predict, so that's why we don't focus on this one as much. So if you get out this paper here, this is going to be the one that you're going to work on that you should have gotten on the way in or that was handed to you. But as you can see here, we have a few things going on. And the nice thing is this table at the top is your key. So you're going to want to focus on these two right here. We're going to look at a type of flower, 4 o'clock flower color. And we're also going to look at human hair. And we're just going to simply do some examples and see if we can make sense of this. Okay, so typically speaking, yeah, this is going to be red. So my drawing is not very good. Let me try using something else. There we go. And this one's going to be white. That is what we see here, and I got those from up here. Okay. My genotypes are going to be determined by my cross right here. So if you look at this cross right here, I'm going to make a Punnett square. So I'm going to do this just like this, and I'm going to do my Punnett square just like that. RW, RW, and the nice thing is all of them are going to be RW. So when it asks what are the genotypes right here, I would simply put RW because it's 100% RW. The best way to describe a 100% is to just put 1.0. Sorry, it's hard to see, but that's what we got here. You could also, I suppose, put 4 to 0, but that's just saying that it's 100%. So either 1 to 0 or 4 to 0. Next, I should say, phenotypes. Well, there's only one phenotype here, just like there's only one genotype. And if we go up here, we'd say, okay, RW gives us a pink. So we would simply write down pink right here. Okay. And same idea as before. Since this is 100%, I'm just going to put 1 to 0 on this one. So that's how we would do that first one. Let's look at the next one. Number 2. Again, we're looking at 2 pink 4 o'clock flowers. So the parents would be RW, and then the other parent would also be RW. So RW and RW. So I would put that over here, RW, RW, because those are our parents here. So make that Punnett square, RW, RW, and I would drop these down, RR, RW, RW, and WW. And so when we look at this here, we're still saying R is still, I take these down here and I say, okay, the genotypes of the parent genotypes. So R and W is just on, it's still, R is saying it's red and W is saying it's white. That's still true. But what are the possible genotypes that I have here? There's a lot of genotypes if you look at these. So there's RR, so I'd write that once, RR, dot, dot. There's RW, there's 2 of them, by the way, RW. And then last but not least, there's WW. This would be a 1 to 2 to 1 ratio. The phenotypes are also going to be drastically different. So we have red, we have pink, and we have white. But guess what? That's also going to be a 1 to 2 to 1 ratio. We simply use this up here to help determine what those are. So that's what we have when we look at this. Let's look at the next example. The next example is going to look at human hair. Typically, when we look at human hair, we know that this is going to be curly and this is going to be straight. So that's coming from right up here. If we have a blend, again, this is going to be the heterozygous is going to be a blend. It's going to be not quite curly and not quite straight. So we're going to take this right here and we're going to make a Punnett square out of this. So we make this just like this. And when we do that, we're going to see that we have the following Punnett square. If we look carefully, we would see there's only 2 genotypes, CC which we have right here. And then we also have CS. Kind of hard to see. I probably shouldn't use yellow. So we have two different types of genotypes. And this, again, since it's a 2 to 2, there's 2 squares and 2 squares. We'll just reduce that down to 1 to 1. Our phenotypes, we would take a look and we'd say, okay, what does CC give us? CC gives us curly. And CS gives us wavy. And again, same thing that we had before. Anytime you have this type of inheritance, the phenotypic ratio is going to be the same as the genotypic ratio, which makes it easy to keep track of. Codominance, again, the alleles are neither dominant nor recessive, and they're both, we say they're both clearly expressed. Oops, sorry, clearly expressed up here. That's the key. This one's a little annoying because we're going to write it. You can see up here, it's a little complicated the way we write it. We write it with these superscripts, which makes it a little trickier. We'll give it a try. So when we look at this here, we're looking at the cow coat. So what that basically, I mean, if you were going to describe a rowan is kind of polka dot. I mean, what you would see is a white cow with red splotches on it. So it really would be, you can Google that later if you want a rowan cow. So our cross is right here. So we're going to go H, R, H, W, H, R, H, R. And we're going to do a Punnett square for this here. Remember, HR is going to be the red, and HW is going to be the white allele. So we kind of do this here. It's going to be kind of tough to do this. My cursor is a little tricky here. So this would be HR, HR. Too bad we couldn't copy and paste, right? This one would be HR, HW. By the way, the reason we're using these subs or superscripts like this is because neither one is dominant. They both show up. So it's kind of like a good way to show it that nothing is dominating the other. So we look at the different genotypes we have here. Different genotypes. Well, I can see here I have two genotypes, HR, HR, and I also have HW, HW. And just like before, it's pretty easy to keep track of this. It is a 1-to-1 ratio, which is nice. The phenotypes we have then would be, okay, so we go up here and we say, okay, where are the HRs? Right here. So that's red. Okay. And then where are the HR, HWs? Right there. So this would be rowan. So this again would be a 1-to-1 ratio. Even though it says, if you look at it fractionally, it's 2-to-2, we can reduce it down to 1-to-1. The next one is also about cows, even though it says a bull, that is a male cow. So if we look at here, the parents we're going to do a red cow. Okay, what is a red cow? Well, we look up here, and that's going to be HR, HR. And then a white bull is going to be HW, HW. If you can't keep track of this, make sure you always use the key up here. And most quiz questions, test questions are always going to have that key up there. And if not, you can usually make it by just kind of walking yourself through the things here. So these would go over here then. HR is the mother, and HW is the father. When it says gametes, that's referring to the Punnett square that we're going to make. So HR. Let me clean this up a little bit here. It's kind of get in the way of things. There, it's a little easier for me. So this would be HR. Where did I get that from? Right up here. And then the bull, the father, is HW. Again, I'm just kind of focusing on what we have on the Punnett square. So the nice thing is all of these are going to be HR, HW. So that's 100%. So all this is going to be pretty easy. Again, this is red. This is white. And this is not a blend. This is not pink. Remember, this is basically like polka dotted cows, and they do exist. We're not making this stuff up. Our genotype is only one, HR, HW, 1 to 0. And the nice thing about this is they are all Rowan, 100%, 1 to 0. And that is number 10. Now, what if these individuals, Rowan individuals, it says, what if we were to cross two of these? So again, I'm looking at these individuals right here, I'm taking them. So they are the heterozygous version of this. So I would say, okay, I got HW times HW. Same thing as this one right here. So I just write that twice. RHW. Write that twice. I'm running out of room. Then I'm going to make my Punnett square here. HRHW, heterozygous. Even though there's no dominant situation going on here, you still have to, terminology still works that way. HRHW, HRHW, and then HWHW. So looking at this here, we have a red. This is fully red right here, right? And then this one right here is going to be fully white. And then let's pick, these guys right here are going to be my Rowans. So I have 3 different genotypes and 3 different phenotypes. So again, even though I know, let me go back here. Yeah, I'll let you finish that one. It looks like I kind of went too far and I can't seem to go backwards on this. No, what I pushed, but that's okay. I'll let you finish that since we did so many examples. And I do want you to finish this page here, which we haven't touched. So on your packet, this would be the second page. I want you to do this one on your own. If you don't know what to do, make sure you look at this key. Same for this. This is the 4th page. Do the same thing I just did, but don't forget to use this this key also. I hope that helps. Finish that worksheet, and hopefully you can get through and make sense of it.